This time we're going to look at some of the factors that can interfere with the enzymes that control the metabolic pathways. So different factors that can influence or affect the enzymes are things such as competitive inhibitors, non-competitive inhibitors, and also something called feedback inhibition. So we'll explore each of those in this video. So starting with the competitive inhibitors, these are competing with the substrate. So a chemical or a substance has a similar shape to the real substrate. What can happen is they bind to the enzyme's active site and thus blocking the real substrate from being able to access its enzyme. So this can slow down chemical reactions because some of the enzymes are going to have their active sites filled with this competitive inhibitor. But this can be overcome by increasing the substrate concentration as long as we make sure there's plenty of the real substrate. Enough of the enzymes will be able to interact with their substrate and thus allow the chemical reactions to still continue. Non-competitive inhibitors, on the other hand, they will bind away from the active site and thus change the shape of the enzyme. So if the active site shape has changed, this means the substrate can no longer bind. So because we've changed the shape of the enzyme, this means that increasing the substrate concentration will have no effect on the inhibitor. We cannot reverse it in this manner. Doesn't matter how much of the substrate is available, the enzymes cannot interact with them, so the rate of reaction will eventually decrease or could stop altogether. So inhibitors may not always be desirable, but it does also give us means and ways of controlling when a metabolic pathway is active within the cell. So something known as your end product feedback inhibition is used to make sure our end products never build up to toxic levels. So in this type of feedback inhibition, the end product is going to act as an inhibitor to an enzyme earlier on in the pathway that generates that product. Okay. So when we've got enough, if we use our example here of isoleucine, that means we don't want to continue this metabolic pathway. So isoleucine will, in that case, act as an inhibitor to enzyme number 1. Okay. So if we take out enzyme number 1, that means threonine, our initial reactant, isn't being converted into intermediate A, which means there's nothing to make B or C or D, and thus we stop producing our end product, isoleucine. Okay. It's acting as an inhibitor to prevent its own production. Thus, it gets used up by the cell, stops acting as an inhibitor, and the pathway can resume once more. So those were our different types of inhibitors. So let's try some past paper questions. So the diagram shows molecules involved in an enzyme-controlled reaction. Which row in the table identifies these molecules? Pause the video and hit play to reveal the correct answer. So the answer for this one is A. So molecule X is the substrate. If we look at molecule Z, we see it has an active site with a complementary shape to X. Substrates always bind to active sites. Okay. Y must be a non-competitive inhibitor. Okay. Because of the shape, it could not enter into the active site, therefore it must bind away. Leaving molecule Z as the enzyme itself because of the active site. So it's important to be able to recognise competitive and non-competitive inhibitors from how they interact with their enzyme, but also from how they will affect the reaction rate. So let's try another. Metabolic pathways can be controlled by feedback inhibition where A. An end product binds to an enzyme found at the start of the pathway. B. An end product binds to an enzyme found at the end of the pathway. C. An enzyme binds to a substrate found at the start of the pathway. Or D. The enzyme binds to a substrate found at the end of the pathway. Again, pause the video and resume when you're ready to reveal the answer. So the correct answer is A. So it's the end product that is binding. So whichever product is made by the pathway is going to act as an inhibitor to the pathway that makes it. So it's got to bind to an enzyme at the start of the pathway in order to shut it down and shut down its own production. So that covers enzymes and inhibitors and metabolic pathways. If you still have any questions or anything needing cleared up, please just let me know.